Welcome back, everyone. So now the next topic that we have is use of biotechnology in the environment, and something that we also know as gray biotechnology. Now, what we find is that this is one of major applications of biotechnology where we have found a lot of different methods that in one way or the other could be very useful for the environment. So let's try to understand all these concepts. So the entire concept of using biotechnology to solve environmental problems is called as bioremediation. Now, when we say bioremediation, the meaning is cleaning up of the environment with the help of living organisms. That's why bioremediation, living organisms solving pollution problems or environmental problems. So that's why it refers to the cleaning of the environment with the help of living organisms. Now, in this particular scenario, when it comes to bioremediation, there are two types of bioremediation. There are two types of bioremediation, one called as in situ bioremediation and the other one called as ex situ bioremediation. In situ and ex situ. In situ means the meaning of the term in situ itself is at the site, and the meaning of ex situ is away from the site. So at the site, away from the site. And that's why when you are trying to treat a contamination at the site of contamination itself, it is called as in situ bioremediation. Whereas when you do the same thing away from the site, it is called as ex situ bioremediation. Now, so that's why in this case you have in situ and ex situ. Now, overall, when it comes to in situ bioremediation, this is where we are trying to solve all the problems wherever the contamination is, wherever the problems are. And in ex situ, we'll take the problem away, the contamination away from its origin, and then do the treatment and bring it back if required. So this is how in situ and ex situ bioremediation techniques are. Now there are various different processes that we use as a part of in situ and ex situ bioremediation. So for example, in case of in situ bioremediation, where it is referring to the treatment of the waste at its site, at the site of contamination. So there are various techniques. We have biosparging, bioventing, bioaugmentation, and biostimulation, etc. Bioculture is another method that is used. So there are a lot of these methods, out of which the most important ones are the first three, or rather the first two: biosparging and bioventing. Now, when it comes to these two processes of biosparging and bioventing, both the methods are pretty similar in their approach, just that there is a slight variation in them while the method is used. So we have biosparging and bioventing. Now, in case of biosparging, we are saying that this is the use of indigenous microorganisms to biodegrade organic constituents in the saturated zone. Whereas when it comes to bioventing, this is where we are making the use of microorganisms in the unsaturated zone. Now, understand exactly what we are saying here with saturated and unsaturated zone. So, suppose if you think of the soil layer, let's say if you have the soil layer like this, below the soil layer, you might know that water layer would start, correct? Water table might start. So, you have the layer of soil up to an extent, and after that, you have the layer of water, correct? Now, in this particular scenario, what we are saying is that there is contamination, right? Whether the soil is contaminated or maybe water is contaminated. Now, soil is at the upper level layer and water is at the lower layer. Now, upper layer, when we are looking at the upper layer, this is where what we see is that upper layer, whether it is upper layer or lower layer rather, microorganisms would be present, correct? Microorganisms would be present in both these layers. So, whether it is the layer of water or whether it is the layer of soil. So water, whether it is layer of water or soil, in both the cases, we know that microorganisms are present. Now, these microorganisms that we have at these places, they would also do biodegradation, correct? They would also do, I mean, they would take care of any contamination. They would start breaking down all the wastes and they would clean up the waste. But that natural process would take a lot of time. So here what we are trying is to make sure that these microorganisms can start working faster. And for them to start working faster, what we need is to provide them extra nutrients. So what we do is that we provide extra nutrients, extra oxygen, etc., so that they can start consuming this nutrition, they can gain energy, and they can work faster. So that's why we are feeding them so that they can start working faster is what we are actually doing here. So in case of whether it is biosparging or bioventing, in both the cases we are doing exactly the same thing, that microorganisms are already present in these areas, just that we want them to start working faster. And to make them work faster, this is where we are introducing some nutrients, or we are introducing some oxygen, and that is leading to these microorganisms working faster. So that's why what we do is that they would be fed. So whether at this layer, they would be fed either at this layer, or they would be fed at this layer. Correct? Either this layer or maybe this layer. So whether it is this layer or this layer, you are sending the food or sending the nutrition and oxygen to these layers. Now sending it to the upper layer is easier, right? Sending to the upper layer is easier. Going down to the water layer is going to be more difficult. Now this layer of where you have water, this is basically called as the saturated zone. This is called as the saturated zone. And the layer above this layer is called as the unsaturated zone. This is called as the unsaturated zone. So unsaturated zone because it does not have enough moisture, and saturated zone because it has enough of moisture. So that's why it is saturated. And unsaturated because it has capacity to engulf more moisture. So that's why saturated and unsaturated zone. And in the unsaturated zone, this process of providing nutrition and oxygen is called as bioventing. This is called as bioventing. And in the saturated zone, when we are doing this, this is called as biosparging. This is called as biosparging. So that's why biosparging and bioventing. Biosparging in the saturated zone, bioventing in the unsaturated zone. And that's why when you are going to the saturated zone, you will have to go at high pressure. You will have to exert high pressure. So that's why the oxygen, when it comes to oxygen and the nutrients, they are injected at high pressure when it comes to biosparging, because you have to go a little down below. Whereas in case of bioventing, we do not need to do it at high pressure. We will be able to do this at low pressure also. So we induce air or oxygen flow at low pressure and also add nutrition to it. So that's why these are two processes which are similar processes, just that there are different layers on land on which these are going. So this is what biosparging and bioventing are. Now, apart from biosparging and bioventing, we also discussed that there is something called as bioaugmentation. Now, this bioaugmentation is a different process where the microorganisms are not already present, but rather we will add these microorganisms to the site of contamination. We have just seen an example before the break. Do you remember? There's an example that we have discussed just before the break, which can be called as bioaugmentation, where you are adding the microorganisms to the site of waste. Can somebody tell me? Anyone remembers? Yes, Gia, you are correct. Vritika, you are also correct. Anyone else who remembers? Hmm. Huh. So this is where we have bioaugmentation, where we this is addition of microorganisms to the site of contamination. To the site of contamination, example being oil zapper. All right. So this is what bioaugmentation is. So overall, that's why we see that these are all the processes that we use, and they all can be called as in situ bioremediation. On the other hand, we have ex situ bioremediation, where we are talking about the transfer of the contaminated material for treatment to some other site. Now, here there are two important techniques, one called as land farming and the other one called as biopile. Now, in case of land farming, what we do is that you would take the soil somewhere and there will be a prepared bed, and periodically the tillage of the soil will be done. Tillage of the soil. So plowing of the field, tilling it. So with the help of tillage, we'll be able to, you can say, stimulate all the microorganisms. The sleeping microorganisms are being woken up so that they can start working faster and they can start working harder. And this is what happens in case of land farming, that the soil will be periodically tilled to stimulate the growth of microorganisms for degradation of this organic waste. So this is what we do with the help of land farming. Similarly, we have biopile. Now, biopile, if you look at the picture right here, what we do is that we prepare the bed, all right? We prepare the bed and we will take care of each and every small property of the soil, everything that the soil needs, whether it is what should be the pH, or should it be acidic in nature, basic in nature, what do we need with the soil? What should be the moisture content of the soil? What kind of nutrients we want in the soil? How much oxygen should be infused into the soil? So each and everything, every bit of the quality of the soil will be taken care of in biopile. And that's why biopile preparation takes a lot of time. It could take 3 to 6 months of time for the preparation of biopile. And that's why just remember that when it comes to biopile, each and every thing with regards to it will be taken care of. So moisture, heat, nutrients, oxygen, pH levels, all these things will be maintained well. And this is what happens in case of biopile. And that's why typically it can take 3 to 6 months of time uh, for biopile to start working. So this is where we see that biopile becomes very, very crucial and very specific in its nature. So there are various techniques we'll use: composting techniques, land farming techniques, all these things, plus also stimulating with the help of microorganisms, with the help of, let's say, even a lot of soil related organisms like earthworms, etc., would also be used. So there are, all these will be used so that we can enhance the quality of soil. So that is called as biopile. So that's why we are first taking it away from the site and then we are preparing biopile here, and then from here on it can either be sent back or maybe it can be sent to wherever we want. Then apart from that, we also have something called as phytoremediation. So as we discussed that when you have phyto, the meaning of phyto is that we are using plants. So that's why when plants are being used for bioremediation, it will be called as phytoremediation. So in case of phytoremediation, we know that there are various concepts that are used. So these are nothing but this is the use of plants to remove contamination from soil and water. And we know that plants in general, you can say that plants are helping in bioremediation, because the basic process of photosynthesis in plants will consume carbon dioxide and give out oxygen. So in a manner they are helping in purification of air. So overall, it's, in a manner you can say that phytoremediation is a concept that is there in each and every plant. Each and every plant is responsible for phytoremediations. But yes, there are certain plants and trees which will contribute more. The example that we have right here is the neem plant, where we know about all the properties that neem plant has. So whether it is antimicrobial properties or insect properties, or acting like a pesticide, there are so many things having medicinal properties. There are a lot of things that neem plant contributes to. So all these things can also be called as phytoremediation, and it will be able to even absorb the poisonous elements and all the heavy metals, etc., from the soil and improve the quality of the soil. So that's why we see that there are a lot of ways in which a plant like neem helps, and similarly there are other plants which can also help you in one way or the other. There are, let's say, even if you think of something like a spinach, even that helps in removal of something from the soil, I mean removal of some of the metals from the soil, and that's why they all can help in one way or the other. A sub process of phytoremediation is phytoextraction. So phytoextraction meaning plants are extracting. How? With the help of their roots, where the roots of the plants will be used to extract everything that is unwanted. All right, extract everything that is unwanted from the plant, and that is called as phytoextraction. So that's why these phytoextraction techniques are also used in a lot of ways, and roots of the plants are capable of phytoextraction, of removing the toxins from the soil. So that's why it is called as phytoextraction, because they extract it away from the soil. And largely when it especially comes to metals, in case of heavy metals, in case of metal toxins, more than microorganisms, plants are more useful, because microorganisms cannot break down the heavy metals easily. They might be able to absorb them, but they cannot break it down, they cannot help in bioremediation. So that is where we see that plants become very very useful in removing all the toxins from the soil. So that's why they help in phyto extraction techniques. And then we have mycoremediation. Now the term myco is always associated with the use of fungus. So when we are making the use of fungus, or as in plural form you call as fungi or fungi or fungi or whatever you want to call, as long as you are using the correct spelling, that is what will matter in your mains examination. So the use of fungus, so that's a fungus is actually a safer term compared to the plural form, where there are so many ways in which people pronounce it and everyone will fight this is the correct pronunciation or not. So mycoremediation, the use of fungus, where they can also help in improving the quality of the soil. They can help in fixing of the nitrates in the soil, and nitrates are the nutrition for the soil. So that's why for the plants rather, so that's why if the soil has nitrates, it will be better for the plants. So that's why in all these cases we see that there are various different techniques which are used in cleaning up of the environment. Read this question, this was asked in your paper. I'm launching the poll for you. Hmm. So 53% of you have marked C, which is the correct answer. Let me discuss this with you. In the context of solving pollution problems, what is or are the advantage or advantages of bioremediation technique? First, it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. Now this is correct. If you remember what we discussed in biosparging and bioventing, what we discussed was that microorganisms are already present. They would have done biodegradation, but very slowly. So, what we want is to increase their pace. We want them to work faster, and hence we have introduced nutrients, hence, we have introduced oxygen so that they can work faster by consuming all this. So, yes, this is correct that it is a technique where we are trying to fasten the process of cleaning of the environment. Second statement says any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganisms. Now, there are two ways in which you can mark this as a wrong statement. Just think of it. First, look at something that we discussed about extreme words. Count the number of extreme words in this particular statement. How many can you count? Can you tell me how many extreme words do you have in this particular statement? Yeah, there are three: any, readily, completely. Three extreme words. With one extreme word, I have told you that read the statement at least twice or thrice. It may be a wrong statement 9 out of 10 times. So just imagine that when you have three extreme words in a sentence, chances are very slim that it could be a correct answer. So, any contaminant with heavy metals such as cadmium and lead. Now, what we discussed about heavy metals is that in case of heavy metals, more than bioremediation by microorganisms, we would prefer plants, right? We would prefer plants. Phytoextraction techniques will be preferred because microorganisms are not very good at breaking down heavy metals. So you can use it. It's not that they are not usable at all. But when you say that any contaminant with heavy metals can be treated readily and completely with the help of microorganisms, that's wrong. That's wrong. Can we do it? Yes, we can do it. But can we do it readily, completely with any heavy metal? No. That's more a job of phytoextraction than of microorganisms. So that's why this is wrong. Third statement, genetic engineering can be used to create microorganisms specifically designed for bioremediation. This is correct. We discussed oil zapper. So a perfect example of statement number three. So that's why statement number three is correct. So correct answer will be C. That is 1 and 3 only. Read this question. A very simple question. Here I need 100% correct answers. Please mark your answers. Right. Still 69% of you have given the correct answer. I don't know where the confusion is. Oil zapper. In what context did we discuss about oil zapper? Did we not discuss this in the context of oil spill? Where do you see in the option oil spill? We do not even need to go to B, C, or D. B says latest technology developed for undersea oil exploration. Nothing to do with oil exploration. Genetically engineered high biofuel yielding maize variety. We have not discussed anything of oil zapper with regards to maize. I don't know why you would mark this as a correct answer. Correct answer is A. Please pay attention to the class. Oil zapper has been discussed at least 4 or 5 times before the break and after the break, and yet there are some of you who have not answered this correctly. This was one of the easiest question to answer after everything being taught. Right. So next topic for us is cloning. But if you have any questions, I mean, from the last half an hour, whatever we discussed, any question? Examples of ex situ bioremediation. uh, we discussed two simple examples. That one is land farming, where the plowing of the field is done periodically, and that plowing of the field, the tilling of the soil, will help in bioremediation. So that is there. Or any prepared soil. I mean, many a times you might also know that you can buy soil. So when you buy that soil, many a times it is biopile, where the soil is very rich in nutrition. So that is also a technique which is ex situ bioremediation. I am from morning class. When should I join to attain the left topics? Uh, I think tomorrow, after the first hour. I mean, after 11 o'clock, we would mostly be at a place where we would have finished everything what we have done in the morning class. So maybe after tomorrow, after 11 o'clock, you can come. Phytoremediation, plants, fruit, flower contain toxin within it. No, it does not go and mix there. It would, I mean, be extracted by the roots, and the concentration will be higher in the roots than the other parts of the soil. And also, these are many of them, most of them rather. There are also some examples which are food fruit bearing or edible crops, but many of them are non-edible crops, or rather non-edible plants, like let's say neem, etc. So then in that case they do not cause any harm as such. But yes, if they are growing in very toxic environments, then uh, the vascular tissues might also take up some of these metals, and that's why the concentration of these metals can be heavy in them. Many a times you will come across some news like this, that some research will be done, or some study will be done on a certain fruit or a certain vegetable, and they find that there the concentration of a certain mineral or a certain uh, toxin is higher. And that happens when they are growing in certain environments like these, or maybe if the amount of pesticides and fertilizers used on them, if that was high, then in that case the concentration might also increase. So is it possible? Yes, it is possible. But that's why in extraction techniques we try not to use something which is edible. Examples of violet biotechnology. See, violet biotechnology, when we say, the meaning of violet biotechnology is that when we are talking about anything related, not related to direct application of biotechnology, but rather we are talking about anything which is um, something like the laws, the acts related to biotechnology, the rules and regulations related to biotechnology, or any paper published on biotechnology, the intellectual property rights related to biotechnology. So all these will be taken as examples of violet biotechnology. Bioreactors basically, you can say that it's any biological medium that can help in transforming something. Let's say petri dishes, etc., that we use, they can be called as bioreactors. Many times the entire cell itself can act as a bioreactor. So all these are examples of bioreactors. Any other question? Metagenome basically is the genome of, you can say, all the organisms taken together, whether it is all the microorganisms or higher organisms, etc. They all can be called as metagenomes and or metagenomic genomics. About tomorrow, science and technology mock test. We didn't complete some syllabus, so should we attempt the test or not? Yes, absolutely, you should. Uh, Anyway, a lot of questions that we have prepared, we have not taken it from directly from the classroom. We have taken a lot of questions from the other topics that I don't teach in the classroom. It just, you can say, it increases the parameter of everything that you're learning. So that's why there will be questions, I mean, a lot of questions which are outside of what we have discussed in the class, and we wanted to do that to increase the number of topics that you are preparing with. And it will have a lot of current affairs also, and some of the current affairs topics that have been in the newspapers on and off, we have taken those topics also. Right. Any question, anything else? Yeah, Ritika, I'm aware of this situation. I already had discussed this with the team, and they said that they'll get it uploaded quickly. There was some issue that happened with the original video that was recorded for economy, and that is where the entire cycle started to run behind schedule. And it has been re-recorded, and now it will be made available for everyone very quickly. And same goes for even the history lecture. I mean, history test recording, which has just happened, and that also will be available very quickly. All right. So let's move on to the next topic, and this is cloning. Now, cloning as a topic also becomes a very important topic because there are some applications that we see of cloning, and India actively has been involved in animal cloning. So, first and foremost, what do we mean by cloning? What's the importance of cloning? What do we mean by this concept of cloning? When we say clone, I mean, when you say that you are producing a clone, the meaning is that you are trying to create an exact replica, right? An exact replica of something is being created, and that will be called as a clone. So in case of animals, when we say animal clones, then in that case we are creating an exact replica, an exact copy of an animal, and that's why it's called as an animal clone. Now in case of cloning, you might, so there is animal cloning that the world has done. There is a possibility of even human cloning. In the films, probably you might have seen that there will be one person who enters a box and another person comes out of another box, and cloning is completed. Unfortunately, in the real world scenario, the cloning process is not that easy. uh, if you have seen Shaktiman or maybe a lot of English films, in all of them, they have shown how cloning can just happen like that. But unfortunately, that's not how cloning works. It's an entire cumbersome process of cloning, and it's a very unstable process as well. Not a very easy process by any means. Now, when it comes to cloning, understand the process of cloning. What we are saying is that it is a process of asexual reproduction. It's a process of asexual reproduction in which the offspring or the progeny is an exact replica of the single parent who has contributed to genetic material. Now, understand this, that when it comes to reproduction, there are two types of reproduction, correct? Either sexual reproduction or asexual reproduction. In case of sexual reproduction, there will be a genetic mixing between the genes of the father and genes of the mother. And then the fertilization that happens between the father and the mother cells leads to the birth of a new offspring. And that's why offspring will have a mixture of genes from mother and father. In case of asexual reproduction, there is a single parent, and that single parent has contributed to all the genetic material that this offspring has. For example, cell division. In microorganisms, in lower organisms, it is seen as a process of reproduction in them. So what happens simply is that the nucleus that they have, the nucleus will divide into two parts, and these two will become nucleus centers, and ultimately they divide into two cells. So from one cell, two cells will be created, from 2 to 4, 4 to 8, and so on. And this process of cell division itself becomes a process of reproduction in these lower animals. Similarly, there is a process called as budding. So in case of budding, what do we see? That you have the organism, a small bud will appear on the body of organism, and this bud starts growing, and ultimately it will detach from this organism, and then become a new individual in itself. So that also is asexual reproduction. Now, that's why when you look at any asexual reproduction, you'll see that there is only one parent involved, and all the genetic material in the offspring is coming from this one single parent. So similarly, in case of cloning also, we are doing the same thing, that we are creating an exact replica of an animal, and hence it is called as animal cloning. Now, when it comes to animal cloning, the first successful example of animal cloning was this. If you know this, this is Dolly the sheep, and this was a sheep named Dolly, and this was the first successful cloning example back in 1997. Now, cloning as a process has been very difficult process. It's not an easy process at all. The success that we got here, I ask this in every class. Do you know how many attempts it took for the scientists to become successful in creating this animal clone? The number of attempts that it took? 5, 10, 20, 1,000. So range is huge. So one correct answer I see here. So Rutika, you have given the correct answer. It took a total of 277 attempts. After 277 attempts, we finally got success. You unfortunately get how much in this examination? Anyway, so 277 attempts. So it has been a very unstable process, a very difficult, a very complex process, and that's why it took a lot of effort. And the effort that it took also has been because animal genes are very different from plant genes. In plant genes, although the process is not that easy, but still compared to animal cloning or any genetic modification in animals or humans, compared to that, in plants, genetic modification is relatively easier because genetically they are not as complex as the animal genes are. And that's why we see that in plants, the genetic modification or any kind of genetic engineering is easier, relatively easier as compared to that in animals. In animals it is way more complex, way more unstable. For example, when we look at the cloning processes that we have seen in India, the first successful example was Samrupa back in 2009, where world's first cloned buffalo was produced with the help of cloning. But unfortunately, did not live for very long, lived for only 5 days and then succumbed to lung infection. Then the second case was of Garima, and this was also 2009. This clone lived longer, lived for almost 2 years, but again, 2011 lost its life. And then the third case was of Gaurav. This was 2016, and this basically was the third example of cloning, which has been fairly better. Um, it lived longer as compared to the first two examples. Recently, there have been a couple of more examples, like in 2020, M29 Murrah bull clones were produced, and a recloning of Hisar Gaurav was done. So these have been examples of animal cloning in India. In 2020, this was done, and recently, last year in March, there was another cloning example, and this was cloning of a cow for the first time. This is cloning of a cow, an indigenous variety of cow called as Gir cow. So this was an example of cloning of Gir cow, and the name that has been given to this cloned cow is Ganga. Reason why did we do this cloning? This cloning has been done as an experiment to enhance milk production. Now, the basic belief here is that when it comes to, let's say, indigenous varieties versus the foreign varieties, indigenous varieties obviously are better suited to the Indian climate, to the Indian tropical climate. So that's why they have better immunity, they have better resilience, and that's why they survive better in these conditions. So instead of always moving towards the hybrid varieties or the foreign varieties, we want that we should focus on the indigenous varieties because these are better suited to the Indian type of climate. And this will something that is maybe in the future it will help in enhancing the milk production. So as an experiment, as a first step towards that, we see that this cloning has been done. So we need to see what how it goes forward and what happens in the future. But overall we see that this is how these are some of the examples of cloning in India. Now what is cloning and how do we do this cloning? The process of cloning, the process of cloning basically starts with a somatic cell. Look at this diagram. We start with something called as a somatic cell, and we take this somatic cell, all right, we take this somatic cell from any individual. Here we have represented a human, it can be an animal, any male or female. We'll take a somatic cell. Now what is a somatic cell? Somatic cell is any non-reproductive cell in the body. For example, in case of humans, you have two reproductive cells. One, the sperm cells found in the males, and then the egg cells which are found in the females. So apart from the sperm cells and the egg cells, every other cell, every other cell is called as somatic cell. All right. So all the non-reproductive cells in the body, they are called as somatic cells. So first, what do? We extract a somatic cell, and from this somatic cell we isolate the nucleus of the somatic cell. All right, we isolate the nucleus of the somatic cell, and once we have isolated the somatic cell nucleus, we take an egg cell, and from the egg cell first we remove the nucleus. We remove or take out the nucleus from this cell, and then we insert the somatic cell. So that's why a somatic cell nuclear transfer happens here. So now you have an egg cell with somatic nucleus. We have an egg cell with somatic nucleus, and then the activation of the egg cell with somatic nucleus will be done. So activation means when we are creating multiple copies out of it. So with the help of electric shocks and chemicals, this egg cell with somatic nucleus will create multiple copies of it. And once we have done that, there are two things that we can do here. Either insert it inside the uterus of a surrogate mother, where embryonic development would start next, or it would be taken to a lab. Sorry, it will be taken to a lab where it can be used for harvesting of stem cells. We'll discuss what is stem cell later. So that's why there are these two types of cloning. One cloning which is called as therapeutic cloning, the other cloning which is called as reproductive cloning. Reproductive cloning is where you want the birth of an offspring. Therapeutic cloning is where all these cells that you have gathered, you want to use it for stem cell or for harvesting of stem cells. So that is what cloning is, as an example. So cloning basically is a process which is performed by somatic cells. Somatic cells, any cell in the body other than the sperm and the egg cells. So that's why any non-reproductive cells. So these are any non-reproductive cells. Any non-reproductive cell will be called as somatic cell. We take the nucleus out of the somatic cell and put it in an enucleated egg cell. Enucleated meaning where you have removed the nucleus. Now, why to do this nuclear removal and replacement? Can you tell me why have we replaced the nucleus? What, in your opinion? Why have we replaced the nucleus? What do you think? Yes, Karan, you have given the correct answer. Shilpa, you've given the correct answer. Dinesh, you have given the correct answer. Ganesh, you have given the correct answer. Yeah, so understand this. Nucleus, what did we learn about nucleus when we started? Nucleus has all the chromosomes. Chromosomes have all the genes. Genes are responsible for all the specific characteristics that you have. So if you remove the nucleus from a cell, you basically have removed all the specific characters of that cell. So from the egg cell, when you remove the nucleus, you have removed all the specific characteristics of that egg cell. Correct? You do not want the mixing of genes. You want all the characters only to come from a single parent. And this single parent is where you have taken the somatic cell from. And hence, the somatic nucleus is what we need. It's like doing the brain transplant. That you have a different thought process, I have a different thought process. You remove my brain and replace it with your brain, I would start thinking how you do. And I would start performing all the activities that you do. So that's like a brain transplant of the cell. So here this is what we are doing, that we are removing the nucleus and we are replacing it with the somatic nucleus, so that we can get all the specific properties of the somatic nucleus instead of having all the specific properties of the nucleus of the egg cell, right? Because we want an exact replica. We do not need mixing of genes, we do not need fertilization to happen. We need an exact replica. An exact replica can only be created when you have nucleus coming from only one single parent, and that is why we are doing this. So why we replace the nucleus, as we want to retain the traits of the single parent only, from where the somatic cell has been derived. All right, so that is the reason why we are doing this replacement. So once we have this egg cell with somatic nucleus, this is where we see that it will be stimulated by electric shock and chemicals, where we'll see that it will become a ball of mass which has approximately 150 unspecialized cells, and this is what is called as blastocyst. This right here, this step itself is called as blastocyst. The undifferentiated bunch of cells is called as blastocyst. And then ultimately, if it is reproductive cloning, this blastocyst from here on will be inserted inside the uterus of a surrogate mother, where the process of embryonic development would start. If not, we'll take it to the lab and it will be used for stem cell therapy, etc. So this is how the process of cloning works. So that's why there are two types of cloning. One where you have reproductive cloning, and the second one which is called as therapeutic cloning. So reproductive cloning is where we want the birth of a new individual, where we want the birth of a new offspring. So then in that case it is called as reproductive cloning. Whereas in case of therapeutic cloning, this is where we use it to harvest stem cells for medical purposes, for medical uses. So that is how we see the difference between reproductive cloning and therapeutic cloning. One simple question that was asked in your examination is this. Very simple question. Please give me 100% correct answers. 91% correct answers. Okay. So if there is any confusion with regards to what we have discussed, What I'm saying is that it is somatic cell nuclear transfer, meaning that we are taking the nucleus from the somatic cell and transferring it. Where exactly are we doing it? We are doing it in the process of cloning. So look at the four options: production of biolarvicides, manufacture of biodegradable plastics, reproductive cloning of animals, production of organisms free of diseases, nothing to do with it. So simple answer, question, the answer will be C. Now coming to the problems that we see with cloning. Now when we look at the problems with cloning, more than technical problems, we have societal problems. And societal problems in a way where we see that there are a lot of changes can happen in the social fabric as it exists today. So for example, when we talk about reproductive cloning, it can undermine society's respect for human life. That we know now that we are able to produce clones, we are capable of cloning, and the medical procedures, let's say, they have been made completely legal. Right now it is banned. It is only to be used in very specific cases, and that too with very uh, pretty much, you can say, prior permissions with from everyone. So then only it is even allowed, otherwise it's not allowed, it's banned all across the world. So now in this particular scenario, if let's say it is allowed, suppose that tomorrow every government just allows that, okay fine, human cloning is allowed, then in that case what happens, that every medical institutions, they can produce human clones, they are capable of doing that. And then it undermines society's respect for human life, because now you know that you can create life, you can create a replica, or an exact replica of somebody. So then in that case it undermines the respect for life, and especially of the clones. You would never be able to see them probably in the with the same respect as you see somebody not born out of natural process of reproduction. So that's one problem. Or maybe they would be treated as a secondary race or even as slaves. We do not know what will happen. Similarly, it also affects the social institutions of marriage and family. That you might even think that why do we even need to get married? I'm good by myself. I can just create a clone of myself and I'll be happy having that child. So why do I need to get married? So there are these things that can bring in a very different change, and it might look seem to be funny right now probably, but it might not be funny when it actually happens. The way it will start to change the social construct of the society is very problematic at times. Then it might also create a global security concern, that it may so happen that today many of the organizations or maybe even countries, they might start working on human cloning, and 10 years 15 years later they can create cloned armies, and then they do not need to send their own armies, because they anyway have no respect for the clones. They've created the clones for this, and then they can start sending them into the battles. All that is possible. Similarly, uh, there is this concept, the concept of designer babies. Now designer baby is a concept where we are saying that we are uh, introducing certain properties at the time of birth, or properties in uh, you can say in the offspring, and these are desirable traits. Now in case of cloning, for example, you might want that instead of going through, let's say, other processes which are available like IVF etc., you might want that, no, I want clone of somebody. I want my child to be the greatest mathematician in this world. So I need to first find the genes of a person who is supposedly the greatest mathematician of the time, and then these genes have to be taken and these genes have to be replicated. So that is what designer baby as a concept is, and we'll discuss more about it later. But this is something that may put more emphasis on infusing desirable traits in the clones. So that's why this is the concept of designer babies, where you are designing the child. You are doing exactly what you want, and you are trying to just give those genes to the child instead of the natural adoption of these genes. One more thing with regards to cloning is to just understand that there is a difference between surrogacy and cloning, because as a process in cloning we know that there are the cells which we are taking from one single parent and we are replicating these cells. But surrogacy is not that. In case of surrogacy, the fertilization is still happening between the male and the female. It is still mixing of the two genes, between the male genes and the female genes. So that's why, as a process, these are not similar, very different. So that's why surrogacy comes under in vitro fertilization, and cloning as a process is pretty different. Then uniqueness of the progeny. We know that in case of cloning you are producing an exact replica, so there is no uniqueness in the progeny. It's exactly having the same genes as the parent. Whereas when it comes to surrogacy, it is still close to what the natural process of reproduction is. It is still reproduction which is sexual in nature, and hence the progeny or the offspring will have genes from both mother and father. So hence uniqueness will be there, because a new set of genes are created with the mixing of the old genes. And then the problem with the lifespan, as we discussed and saw some of the examples also, that cloned animals have not led a long and healthy life, not a normal life by any means. We always have to keep them isolated with a lot of medication, etc., to ensure that they do not die, they do not succumb to some kind of infection. So there is a lot that we have had to do to even ensure their survival. But that's not the case with surrogacy. In surrogacy, the child born out of surrogacy is completely normal, and there's no issue whatsoever that we see there. And then there are ethical issues also, which are pretty much raised with regards to cloning, with regards to in vitro fertilization, or in case of surrogacy. These issues are not really laid, and anyway, because there are rules and regulations that already are in place to ensure that there is nothing that goes wrong. So that's why there are ethical concerns largely with cloning, and as we discussed, all the concerns that we have with human cloning, and that's why there are so many problems. And at the same time, even if you don't consider those problems, it's also about the misuse of the embryo. The embryonic development may not happen because you are using it for some kind of research or some kind of study. So then in a manner you are also killing that embryo, and killing an embryo is equivalent to killing a life. The same embryo might have taken the shape of a life later, but you have just used it for research purposes and you probably have killed it. So that's why many cultures would believe that it is equivalent to killing a life. So that's why there are ethical concerns, there are moral concerns that are raised with uh, cloning. And again, these are not concerns that are largely raised with surrogacy. So that's why you need to understand the basic differences that we have in all these scenarios. Any question, anything related to cloning? We are using somatic cell nucleus for cloning because it contains 46 chromosomes, and not using reproductive cells because it contains only 23 chromosomes. No, that's wrong, because inside the nucleus there are 23 pairs. All right, there are 23 pairs. When the mixing happens, when the genetic mixing happens during reproduction, half of it is taken from the females and half of it taken is taken from the males. And if, let's say, you had let the nucleus be there and you introduced a new nucleus, then in that case again that mixing would have happened between the two nuclei. So individually, every nucleus will have 23 pairs. They cannot have half the pairs. How egg created? Egg cell will be already present inside the body of the females. It's not created artificially as such. What are the main issues with cloning? The main issues we discussed, I mean, they are practically the problem. Whether you talk about the issues of how it can be misused, it can be misused for any purposes, that people will start thinking of how to create slaves, or maybe a market can be created out of clones. And at the same time, we may see that it will structurally also change the society, the respect that we have for human life, because we know that we can create clones. So tomorrow people might look to create clones of all their near and dear ones. So there are a lot of things that can go wrong in this case of cloning? Indian society not agree to cloning. So see, I mean, it's not only about Indian society. I mean, any society wouldn't want the social fabric to see change in that sense. So that's why the ban is not only in India. The ban is there all across the world. I feel good in ISRO and space class. Your choice, but unfortunately, you have to read everything. Right? Any other question? Can we move ahead? If cloning fails, do they become zombies? No. Uh, there can be genetic mutation. They might have genetic issues. Uh, can't call it as zombies. That's a very different concept altogether. Hmm. So moving on, the next topic that we have is stem cells. Now, stem cells basically are very unique concept where we are talking about the origin of the cell and how we can use these cells because of the way they are. Now, stem cell is where all the cells originate. We have different types of cells in the body, correct? You have the white blood cells, red blood cells, nerve cells, skin cells, muscle cells. So there are different types of cells in the body. Now, in all these kind of cells that we have, they have their different functions. So they have different functions, different shapes, but the origin of all these specialized cells is the stem cell. They all stem from this particular cell. That's why these are stem cells. This is where they begin from. This is their origin. So they all start from stem cells, which are nothing but unspecialized cells. Stem cells do not have any particular property, they do not have any characteristic, they do not have any function. They are just unspecialized cells. And from these unspecialized cells, we see that we now start getting all these different specialized cells. Now, this becomes very, very crucial. How it becomes crucial, we'll discuss that later. But what we understand is that because of this unspecialized nature of the cell, this is extremely important for us in medical science. So stem cells are nothing but the origin, right? They are the origin of any cell. Just think of an equivalent example can be an embryo, where the embryo starts to develop into a child, correct? So at the time of the embryonic stage, let's say after fertilization, after 10 days, 15 days, 20 days after fertilization, is just an embryonic stage. uh, there is no hands, legs, heart, brain, etc. All the organs have not developed yet. The organs will start developing, and by the time the child is ready for birth, 9 years into the womb, this is where the child would have now developed all the different organs in the body. So now, so similar something we see in the case of cells also, that cells, they start from a stem cell, where the stem cell is completely undifferentiated, unspecialized, and then finally it converts or specializes into all these specific cells. So stem cells become the origin of every cell. All right, stem cells, in a manner you can say that these are the origin. Stem cells are the origin of every cell, every functional cell. All right, stem cells basically origin of every functional cell. Now, in case of stem cells, what we see is that before stem cells they start to specialize, they would first create a lot of copies of themselves, meaning that they would undergo multiple cycles of cell division. They'll undergo multiple cycles of cell division, and then they would finally start specializing. So that's where there are, when it comes to stem cells, we know that stem cells become very crucial because they are the raw material for all the other body cells. And these stem cells, they can just convert into anything, they can specialize into anything. The same stem cell can specialize into a red blood cell, the same stem cell can specialize into a white blood cell, the same stem cell can specialize into a heart cell, into a muscle cell. So the same stem cell can convert or specialize into a lot of different types of cells. All right. So that's why we see that stem cells become extremely important in that particular scenario, because they help in all these different ways. So that's why stem cells become very crucial, because they would be useful in creating all these different cells at the same time before they become specialized. That's why stem cells become very crucial. So now, when it comes to these stem cells, there are two important properties that they have, which we need to understand. One, their ability of self renewal, and their ability to specialize. Meaning that first point means that they would undergo through numerous cycles of cell division while maintaining their undifferentiated state. So what happens is that from one stem cell to 2 to 4, 4 to 8, 8 to 16, 16 to 32, and so on, they would keep on creating multiple copies of themselves before they start specializing. Ultimately, they would start specializing, but before they start specializing, they would undergo multiple cycles of cell division. All right. So then in that case, you have a lot of stem cells available for your usage. That's why this becomes a very important property of stem cell. And then their ability to specialize, because we know that these stem cells can convert or specialize into a lot of different types of body cells. So hence they will be very, very useful in that sense. They would be very, very useful because they can create or they can convert into so many different types of cells. So that's why their ability of self renewal and their ability to specialize, these two abilities, these two properties, they become extremely important for us, and that is why we see so many applications of stem cells. Now there are different types of stem cells depending on their functionalities, depending on their properties. There are different types of stem cells. We have totipotent, pluripotent, and multipotent cells. All right, so these are the three types of stem cells. So totipotent cells, pluripotent cells, and multipotent cells. Now understand what they are. What we do we mean by totipotent, pluripotent, and multipotent? Totipotent cells, toti in a manner you can say is derived from the word total. So that's why totipotent. Why totipotent? Because these are the cells, these are the stem cells that have an ability to specialize into any kind of cell in the body. Any kind of cell. They can specialize into all cell types in a body, all cell types in the body with addition of the extra embryonic or placental cells. So that's why they can just specialize into any body cell, any kind of cell that we find in the body. Now, these totipotent cells, they can be derived just after fertilization. As soon as the fertilization has happened between the male and the female cells, the first two cycles of cell division after fertilization is where you find totipotent cells. Just as the cell division has started after fertilization, so 1 to 2, 2 to 4 creations have started only in the first two steps. The first two cycles of cell division is where we find totipotent cells. So that's where they become very, very crucial. So they basically are, that's why they are also called as embryonic stem cells. They are called as embryonic stem cells. All right, they are called as embryonic stem cells, and they have an ability to specialize. They can specialize into any body cell. All right, they can specialize into any cell in the body. So that's why they become very, very crucial, because they have unlimited capacity to specialize. So only during the first two cycles, only within the first two cycles of cell division after fertilization is when you have totipotent cells. All right, then we have pluripotent cells. Now in case of pluripotent cells, even they are also embryonic in nature. These are also embryonic stem cells only. These are also embryonic stem cells, but with a difference, that these are stem cells after the first two cycles of cell division. All right, they are after first two cycles of cell division. So they also have a wonderful capacity to specialize. They can also specialize into all the cell types, but not into extra embryonic or placental cells. So in a manner you can say that they can specialize into they can specialize into almost any cell. All right, they can specialize into almost any cell. They cannot specialize into every cell, which was the case in totipotent. In multipotent, almost every cell they can specialize into, and they are also embryonic in nature. This is also during the time of the embryonic development. Now we have the multipotent cells or multipotent stem cells. Now these multipotent stem cells, these are also called as adult stem cells. These are adult or also called as perinatal stem cells. Adult or perinatal stem cells, meaning that these are the cells which are found after the development of embryo into a child has happened. After the development, when all the organs have grown, all the legs, hands, etc. have grown. So at the time of the birth or after the birth, adult stem cells are after the birth. So adult does not mean that you have to be 16 years of age or 20 years of age. No, after the birth they are called as adult stem cells. So adult stem cells where all the you can say organs have grown and they have specialized already. So in this particular scenario we see that they have a limited ability to specialize. They have a limited ability to specialize. They cannot specialize into anything. They can specialize only into the cells which are found near their location. Meaning that if you are deriving stem cells from an area near the heart, then these stem cells can convert or specialize into heart cells or the other muscle cells or nerve cells which are found in this area. But they cannot specialize into brain cells. They cannot specialize into anything else. They would become limited in their ability to specialize. So that is what are called as multipotent cells. So totipotent cells and pluripotent cells, they have almost unlimited capability to specialize. They can specialize into all the different types of body cells. But in case of multipotent cells, they do not have that capacity. They have a limited capacity to specialize because the body already has grown. The body already has grown and all the specialized organs and body parts are already present, and that's why multipotent cells have a limited ability to specialize, right? So here in this case I'm just writing it here in this explanation. They have, sorry, they have a limited ability to specialize as the body or as the body organs and tissues have already grown completely. All right, so that's why they have a limited ability to specialize. So that's why these are the three types of stem cells. Now from these all stem cells what we see is that all these stem cells, they can be used in various ways. Now this is where all these properties will come into the picture. So for example we have regenerative medicine. So regenerative medicine is where we are trying to regenerate damaged tissues or organs with the help of stem cells. Now how to do that? What we do is that whatever be the injured part, or could be any reason, could be an accident, could have been even a heart attack. That when heart attack also happens, many of the muscle cells, they basically they get damaged. So these damaged muscle cells can be replaced with stem cells, and these cells will just grow into those muscles. they'll just grow into those muscle cells. Similarly, for an injured part of the body also, stem cell can be used, and it can regenerate that body organ that has gone damaged, that has been damaged. So this is where their ability to specialize comes into the picture, because now, because of their ability to self renew, we have multiple copies available, and because of their ability to specialize, we know that they can specialize into these muscle cells, or nerve cells, or any kind of cell that we needed. So that's why stem cells become very crucial in regenerative medicine. Similarly, they can also help in drug testing. They can also help in drug testing. How? What we can do is that we can grow tissues or organs out of stem cells and do the drug test or the vaccine test on that before releasing it. Before releasing it for clinical trials or even animal trials, the first step can be to test the same drug or the vaccine on stem cells, or the organs and the tissues grown from stem cells. And in that sense, we'll be able to use them better. Correct? We'll be able to understand how the drugs are working, or if there is any negative impact that we see on this organ or the tissue. Depending on that, we can take a decision. So that's why in drug testing it becomes crucial. Then it can help in disease modeling. Now, disease modeling basically is where they we try to understand the mechanism of disease development, that what exactly is going wrong, when is it going wrong. Let's say there is a particular cell which is getting affected due to some disease. We can study at what step during its specialization from a stem cell into a fully functional cell, where are the things going wrong. If we are able to identify what went wrong, we can make the use of this information to cure the disease. So that's why stem cells can be used in disease modeling, where we can try and understand by just observing the growth, we can try and understand what's going wrong. Then we obviously know about stem cell therapy, where we are talking about various different conditions like immune disorders or leukemia, thalassemia, lymphoma, damaged cornea. There are all these cases where we can make the use of stem cells. So all this will be called as stem cell therapy, where stem cells are used in the medical procedures where you do not have normal medical procedures, or let's say medicines or vaccines available against that disease. Then we have the use also in basic research, that we can use it just to understand the biological processes, how the cell division works, how the differentiation works, how the specialization works. So we can just use it for all that basic study and research as well. Then there is tissue engineering, the concept of tissue engineering, where we want to make the use of stem cells to grow tissues out of stem cells. And this is again specifically important in terms of organ transplant, or whenever, as I told you, the accident cases where some part of the body might have become damaged. So in all these cases also tissue engineering can be used, and it's an active field of research where we see that the stem cells are going to play a huge role. And then they can also be useful in neurodegenerative diseases, where diseases like Parkinson's or Alzheimer's, where some nerve has been damaged, and hence new neurons can be created with the help of stem cells, and they can be used to replace the ones that have been damaged or lost. So that's why in case of neurodegenerative diseases, we can use them. Similarly, we can also use them in autoimmune diseases. I mean, all these examples, these are just some examples, and there are a lot of other examples where we see the use of stem cells. Autoimmune diseases, where immune system attacks itself, or the immune system attacks the body. So that's why immune system becomes weak, and since the immune system itself is not responding, itself is attacking the body. So then it becomes very difficult for the body to uh, fight against any disease. And this is where we see that again stem cells can be utilized to cure autoimmune diseases. Then there are even dental applications. They can be used in dental applications as well. So there are multiple ways in which stem cells can be used in one way or the other. Then lastly, there are certain problems, there are certain issues that we have with stem cells. So, for example, there are ethical issues in the use of stem cells, ethical issues. So, ethical issues where we are talking about the embryonic stem cells, the use of embryonic stem cells, because we know that embryonic stem cells have kind of an unlimited power to specialize into something, but we do not really want to go there, right? We do not really want to go to a situation where it can lead to a problem altogether, and it can lead to commercialization of the embryo, right? It can lead to commercialization of embryo, which can become very very dangerous, because embryonic development will only happen so that it can be sold in the market and embryos will be utilized for stem cell harvesting. So we do not want to go there, and we do not want these things to happen. So that's why embryonic stem cells is something that we do not really want to use for stem cell therapy. Although they are good, they have a better uh, chance of getting medical therapies out of it. Yet, because of these ethical problems, we do not really want to actively use the embryonic stem cells. Problem with the use of adult stem cell is that it has a limited ability to specialize. That, as I told you, that they become location specific, right? They become location specific, meaning that depending on where they are located, they can only specialize into the cells which are in the vicinity of that location. So that's why, because of that limited ability, they cannot be used as well as the embryonic stem cells could have been used. So that's why, on one hand, you have ethical problems because of which you do not want to use embryonic cells, and on the other hand, you have adult stem cells which have a limited ability to specialize, and hence they are not as useful as the embryonic stem cells are. Now, there is, you can say, a solution to these two problems, and this is called as induced pluripotent cells. Induced pluripotent stem cells. So, induced pluripotent stem cells basically are the kinds of stem cells where we convert, this is conversion of adult stem cells into embryonic stem cells. So now we have been able to even find methods in which adult stem cells can be converted into embryonic stem cells. So that is basically called as induced pluripotent stem cells. All right, these are called as induced pluripotent stem cells. So that's why you can say, in a manner, it's a solution between the two problems that we just discussed. Read this question. This was asked in your paper. I launched the poll for you. Please mark your answers. Wonderful. 83% of you have marked the correct answer, which is B. Now, question says, with reference to stem cells frequently in the news, which of the following statements is or are correct? First statement says, stem cells can be derived from mammals only. An extreme word. So we have to be careful. This is wrong, because it's not only mammals, but you can say higher plants and animals. Higher plants and animals, usually they have stem cells. So in animals, you understand, in animals it is very similar to what we discuss about humans and everything that we see in humans. But in case of plants also, even plants have stem cells. Plants also have stem cells. For example, there are meristems or meristematic tissues. So meristematic tissues basically is where you have all the stem cells. In a way, you can say these are the tips of the plant. Tips of the plant, basically all the tips, whether it is the tip of the stem from where the stem might grow, or the tip where from leaves are growing, or where flowers are growing, or even in case of roots. So wherever you have a tip in the plant, all these tips basically have stem cells, because this is where the new growth is happening from, and hence you have stem cells even in the plants. So it's not only uh, that's why that's why it's not only the stem cells from the perspective of animals, not only animals or humans, but also from the perspective of plants. So that's why statement number one is wrong. Second statement says stem cells can be used for screening new drugs. We discussed that for drug testing you can use it, even for vaccine development this can be used. Vaccine testing also it can be used. So two is correct. Second, the third statement is stem cells can be used for medical therapies. We discussed about stem cell therapy. So yes, this is also correct. So two and three, these are the two statements which are correct statements. So any question if you have with regards to any every discussion that we have done in stem cells? Can we harvest adult stem cells at any time of lifespan? How much time will it take to develop organs from stem cells? So overall, let's say when it comes to harvesting, harvesting can be done at any stage. It can be done from a newborn child if you want. It can be done from a 10-year-old, 20-year-old, 30, 40, 50, 60-year-old person. You can do that. Bone marrow and the fat cells in our body, they contain uh, the stem cells, and that's why harvesting can be done from there. And yes, at any stage in life, theoretically it is possible to do that. You had another question, I think. How much time will it take for the development of the organ from the stem cells? The usual process will be similar to the process that we anyway have for organ development. The time that it takes for the time that it takes for the development of the organs from in the body, it will take similar time. So there is no unusual time period that we have. The cell division will happen at its normal pace. How it can be used, I Vikalp, I'm not sure what exactly the context is. Acha, in screening drugs. So in screening of the drugs, basically what we do is that first we'll take the stem cells and we'll grow the organ out of the stem cells. Now, uh, when it has grown, stem cell has grown into a tissue or has grown into the organ, this is where we'll inject the pieces of the medicine or the vaccine, and we'll see what happens to that organ, and we will make a let's say a functional organ out of it, and we'll try to test it on it. Which type of stem cell is more powerful in these three types? I mean, technically, if you go by their ability to specialize, totipotent cells have the best ability to specialize. That's why we said that they can develop into any kind of cell in the body. So that's why they, in that case, I mean, if you are strictly speaking from the perspective of our use, you can say that they are the ones which probably will be the best for us. Can clones be created from women? Yes, I mean, you can do the cloning of males and females. How will it be used in studying the development of a disease? So basically, in the development of a disease, what we do is that we study. So let's say the stem cell starts to grow, and normally if the stem cell is growing into an organ, it would take like 2 months of time, usually almost up to 2 months of time, for the stem cells to grow into an organ. Now then there is a normal method of stem cell into growing into an organ, and then there is a disease where we see that there is something wrong in it. So what we do is we study how exactly the growth is happening at each and every step, and that will give us an idea about how exactly the growth, where exactly in the entire growth process is the cell going wrong, where is it lacking, what is going wrong. So all that can be done when we study the normal growth of stem cell into cell plus the diseased growth of cell into uh, stem cell into a cell. Why stem cells have limited ability to specialize? It basically happens because of the growth, because in the embryonic stage the development of the organs have not happened. So right now that's why they have all the ability to grow, because they are about to grow into everything. But and that's why they have the genes which are responsible for all these growths. But in case of the adults, those genes are now have become specific to that organ or specific to that place, and that's why the stem cells also do not have that stability anymore. Germ cells like sperm and egg cells contain 23 chromosomes. Yes, they have these pairs. Do you have any complication of terms related to biotech prelims? I mean, these terms keep on appearing in the paper. I mean, your newspapers from time to time, but largely uh, this time I don't really remember. I mean, I can't recall that something that I would have read in the last 6 months that I can say is a complicated term. Uh, but yeah, I mean, there must be something that I'm forgetting, and uh, we'll see that. We'll take current affairs classes later anyway, before the examination. We'll do a revision of all that. Anything else? Any other question? How long does totipotent cell can be stored? So see, storage will be very similar to even how the storage of blood happens, and how long we can even uh, store blood. So generally it would, you can say, you can give or take 1 month to 45 days of time for them to be stored. Induced pluripotent cells mean, so induced pluripotent cells basically mean that we are converting the properties of adult stem cells into the properties of embryonic cells. So that's why induced pluripotent, because we are inducing the properties of pluripotent cells in the adult stem cells. When will the class end? You can leave if you want. Nobody has stopped you. Class will end probably in the next 5-10 minutes. Is stem cell Legal in India? Yes, I mean there are procedures for which stem cells are used even in treatment of cancer and AIDS. Today we have started using stem cell therapy. So of course the use is legal, but yes, there are limitations wherever the things can go wrong. There are limitations to the use of stem cells. Email ID I'd given it before also. It is rashesh.kumar@byju's.com. Totipotent and pluripotent difference. The difference basically is that totipotent is just after fertilization. As soon as the fertilization between the male and the female reproductive cells have happened, just after fertilization, when you take out the stem cells from there, these are totipotent. And after the first two cycles of cell division after fertilization has happened, then the ones that you have are pluripotent cells. How can disease be treated by stem cells? So see, in the treatment of stem cells, what we basically do is that uh, either it is replacement, that the cells which are diseased, they will be replaced by stem cells, and these stem cells will then start to grow, or we grow a tissue or a cell out of the stem cell and then we replace it in the body. So it's largely about the replacement. So that is basically what we say in terms of uh, these stem cells. What oligopotent cells? So oligopotent cells, you can say in a manner they are very similar to what multipotent cells are, although in their ability to differentiate and ability to specialize, they're even more limited in that sense, right? So you all had a lot of questions from stem cells. Anything else that I'm missing? Any question that you have? Right. So in that case, let's stop here only. We can start with DNA fingerprinting in tomorrow's class. So there's a small portion of this chapter which is left. So maybe we'll take approximately like 45 minutes to 1 hour to complete this chapter, and then we'll start with our next chapter, which is nanotechnology. So we'll complete this chapter. We'll begin with nanotechnology, and then after that, robotics and artificial intelligence will be the next two chapters that we are going to discuss, right? So please be back tomorrow at 10. We'll start with the session. We'll start at 10 o'clock and we'll go on till almost the same time, 7:38 p.m., depending on the topic and what we are able to finish. How do we induce the properties of embryonic cells on an adult stem cell? It's a completely technical process. I don't think it's even something that we can discuss here. It's a, a large, complicated process that we use in making those genetic changes in the adult stem cells. All right. So this is end of today's session. Thank you very much for being here. Please, I don't think you will have enough time to revise between the sessions, but at least after tomorrow's session, please go back and revise everything. Thank you.